Have you ever wondered what goes on inside the brains of animals? How do they perceive the world, process information, and react to stimuli? These are some of the questions that neuroscience tries to answer by studying the structure and function of the nervous system. But studying the brain is not an easy task, especially when the animals are moving and changing their shape. Imagine trying to track the activity of hundreds or thousands of neurons in a worm that is constantly twisting and turning. How can you keep track of which neuron is which and how they are connected and communicating with each other? This is where artificial intelligence, or AI, comes in handy. We will talk about a new AI method that can track neurons in moving and deforming animals, such as worms, using deep learning. This method was developed by scientists from EPFL and Harvard, and it represents a significant advancement in neuroscience research as it enhances brain imaging efficiency and deepens our understanding of neural circuits. The method that the scientists developed is based on a convolutional neural network, or CNN. This neural network consists of multiple layers that can extract features and patterns from the input data and produce an output that corresponds to the desired task, such as classification, segmentation, or detection. The task that the CNN performs in this method is to track the neurons in the brain of an animal, whether they are represented as points or volumes, and assign them unique labels that can be used to identify them across different frames and postures. The input data for the CNN are the images of the brain, which are obtained by using a technique called calcium imaging, which measures the changes in the fluorescence of neurons when they are active. The CNN learns to track the neurons by using a technique called targeted augmentation, which automatically creates reliable annotations for new postures, reducing the need for manual labeling. Targeted augmentation works by applying random transformations, such as rotation, scaling, and translation, to the images of the brain, and then using a registration algorithm to align them with the original images and transfer the labels of the neurons. This way, the CNN can learn from a variety of postures and scenarios and generalize better to new and unseen data. The CNN also uses a technique called self-attention, which allows it to focus on the most relevant parts of the input data and ignore the irrelevant or noisy parts. Self-attention works by computing the similarity between different regions of the input data and assigning weights to them according to their importance. This way, the CNN can learn to track the neurons more accurately and robustly, even when they are occluded or distorted by the movement of the animal. The results of the method were impressive and promising. The method was tested on the roundworm Caenorhabditis elegans, which has a large number of neurons in its brain and is a widely used model organism in neuroscience research. The method was able to track the neurons in the worm with high accuracy and efficiency, even when the worm was moving and deforming in different ways. The method was also able to track the neurons in other animals, such as zebrafish and mice, demonstrating its versatility and applicability. The method was not only able to track the neurons, but also to reveal complex neuronal behaviors and responses to stimuli, such as food, light, and touch. The method was able to capture the dynamics and interactions of the neural circuits and how they changed over time and space. The method was also able to identify the functional roles and properties of the neurons, such as their activity patterns, firing rates, and connectivity. The method was able to provide a comprehensive and detailed picture of the neural activity in the brain of the animal and how it related to its behavior and environment. The method also has implications for AI research, as it showcases the power and potential of deep learning for solving complex and challenging problems. The method also raises some interesting and important questions and issues, such as the ethical and social implications of using AI for studying the brain and the limitations and risks of relying on AI for making decisions and judgments. In this video, we have talked about a new AI method that can track neurons in moving and deforming animals. We hope that you have enjoyed this video and that you have learned something new and interesting. If you have any questions or comments, please feel free to leave them below. We would love to hear from you and get your feedback. And if you liked this video, please give it a thumbs up and subscribe to our channel for more videos like this. Thank you for watching, and see you next time.